Pronto, o Márcio vai dar liberar. Hello, everybody. So, good afternoon. It's our pleasure to receive Dr. Leonardo Oliveira Mendonça here for today's immunowebinars. So, Dr. Mendonça is from the Faculty of Medicine from the University of São Paulo. He's the president for the Brazilian Consortium of Inborn Errors of Immunity. So he's going to talk to us about inborn errors of immunity and universe in expansion. So thank you very much, Dr. Oliveira. The floor is yours.
So I'd like to thank you for the invite, for being here today. I'm a physician and I work in the border of immunology. Tá, claro. And I work in the border of the clinics with the laboratory. So I do part of my life in the clinic and part of my life in the laboratory. And I have been collaborating with Professor Alessandra Pontillo for many years here in the ICB. I have already been here for several times doing some experiments in her lab. And it's a pleasure to come back here today to talk about this expansion that we had observed in the group of inborn errors of immunity. So what can I... Yes, so I have been invited to talk about the expansion that we observed in the inborn errors of immunity over the last years. And we also observed the same augmented spectrum of the disease here in Brazil since we started this project that is the CNA3E, CNA3E, which means the National Center of Inborn Errors of Immunity and Immune Dysregulation. So it's the part of the spectrum of the group of natural disease, natural genetic disease that has bring this expansion to the inborn errors of immunity, to the group of inborn errors of immunity. So I'm going to start with this clinical picture. That is, this guy is a four-month-old guy with a sterile fever and this skin lesion, severe skin lesion, arthritis with osteomyelitis that has came to our attention several years ago. And the physicians were questioning themselves, where was the infection, why it was developed so severe in the systemic disease. And we started studying these patients in 2019 because of this severe phenotype in which we could not find any infectious agent responsible for the phenotype. So we started questioning ourselves if this was really infective, if there was an infectious agent here in this patient, and we started to look differently for the patients that we were investigating at that time. So for the first time, we were questioning ourselves if this phenotype that resemble infections was really infection for the first time in the inborn errors of immunity community. It was in 2019 this patient we first seen this patient. So since then, not just us, but all the groups of physicians all over the world that studies inborn errors of immunity, they started looking differently to these patients and looking to a different phenotype that was found for this group of patients. Because until 2017 and 18, we only thought about genetic determined disease when we're looking to recurrent severe infections. We just thought that genetic disease or genetic inborn errors of immunity could be related to severe infections. And when we started observing that also sterile inflammation, autoimmunity, lymphoproliferation, severe allergies could also be a genetic mediated disease, we started to reclassify this disease. But it was not just in the phenotypes that we were observing this expansion in the group of inborn errors of immunity. Just because of this phenotype observation, we also observed that we needed different technologies to investigate these patients. And this is the main reason why we had so many several diseases over the last years, because we observed that we needed a target, not just target genetic sequencing, we needed whole exome sequencing, we needed whole genome sequencing, we needed larger genome sequence here to investigate these patients. We started to observe that different mechanisms were involved within the same phenotype or within different phenotypes, and this was one of the reasons why we cataloged this disease in different groups, and I'm going to show you after this here. And we started observing that we also needed to change the geography of the genetic sequencing, especially because to align our sequencing with European databases or North American databases was not enough for the genetic analysis of our patients. So these four reasons were the main responsible for the expansion in the inborn errors of immunity here in Brazil. So I'm going to start first with the immunodeficiency concept to the change to inborn errors of immunity concept. So until 2017, as I said, we all recognized this disease as immunodeficiency because we only thought there is curiosity because at the time, we thought that loss of function was the responsibility or was responsible for all inborn errors of immunity. It's easy to link the loss of some function with the augmented infection risk or infections of something. But there was a duality in what we found, that is the loss and gain of function is responsible for the same phenotype sometimes. So this was very important for our field because it was responsible for the clinical expansion of from where we act, from where we see patients, or the way we see these patients, because this really improved our recognition of some disorders. So the first inborn error of disease described at that time was in 1952. There was just the number one inborn error of immunity is the Bruton gamma globulinemia. It's the patient with the complete absence of B cells without the production of antibodies. And now we have 559 genetic determined inborn errors of immunity. So in over 15 years, 60 years, we had an expansion of several numbers of genes discovered. And especially in the last 2 years, we also had found, we also categorize, or we also classify a group of diseases that are not inherited, are not inborn, because we recognize now some groups that we call phenocopies that are disease linked or to somatic mutation, which is not transmitted, so it's not inborn, and anti-cytokine that I'm going to talk after. So the way we physicians see inborn errors of immunity changed a lot in the last year, because initially we just saw it as recurrent infection, but now we understand that these patients or this group of disease also has a predisposition to autoimmunity. We see these patients in diverse clinical scenarios like cytopenias, enteropathy, gastroenterology, endocrinopathy, and organ-specific disease. We see these patients in autoinflammation also, so sterile inflammation, as the patient that I showed before, serositis, sterile osteitis, or neutrophilic dermatosis. We also see inborn errors of immunity here. We see these patients also in severe or refractory allergy, so patients with severe eczema, eosinophilia, and food allergy. Now we also recognize these patients as genetic determined disease, and we are growing this number also here in malignancy. By now, we have a small number of patients with clear predisposition to malignancy, but we are studying, for example, in our group, the lymphoproliferation phenotype as a specific manifestation of this disease. So it's very important to observe the number of the clinical scenarios in which we see inborn errors of immunity now, and this was responsible just because of the change of the name of the field to immunodeficiency to inborn errors of immunity. Here I have an example of CTLA-4 haploinsufficiency, which in just one phenotype, on one disease, on one genotype, we can have autoimmunity, we can have infection, we can have lymphocytic infiltration in central nervous system or in other organs, and we have some patients with allergy and also with infection. So in just one gene, we can have all the phenotypes that we are talking here about immunodeficiency. And why study humans? So there is a very important point when we change from the mouse knockout to the human disease, to the human genetic disease. So these patients or these patients that we are studying in this project, they are the clear phenotype of a mouse knockout. So we see in patients a natural error of the nature. So these patients come to us with the clear loss of function or gain of function mutations, and we can observe all the phenotypes of interest that we would like to see in the mouse knockout, but we see in these patients. What is very interesting, especially here in Brazil, is that there is a possibility to find new models of knockout because of that mixture we see in the population of Brazil. There is a very important or a real important actual risk of finding new mutations or new genes related disease that was not observed in other parts of the world. So it's very important for us to keep a look or to continue interpret and understand these patients here in disease. And we have look into one gene, but different pathways and different learns that we saw over the last year. So this is a patient from Goiás that we have seen some years ago. He has, here you can clearly see that he's almost a fungus man, so he has fungus in all over his body. And we were studying what could lead to this fungus predisposition, because also in the skin, we had a biopsy that was compatible with lupus. So in the same patient, we had this duality of being like predisposition to some fungus infection and also to the predisposition of autoimmunity. So the same gene that this patient has a mutation in the STAT1 gain of function, we also observed the deficit of the FOXP3 cells, and we also observed the predisposition or the formation of autoantibodies. So a single patient has taught us many things about how the phenotype and the genotype and the mechanisms are involved in several aspects of human disease. And all through, we see this expansion in the inborn errors of immunity in numbers. We have to be clear that in the next, probably in the next 20 or 30 years, we're going to see more than 1,000 genes involved in inborn errors of immunity. So here is the classification of inborn errors of immunity. So up to the last classification in 2024, we had 500 genes involved in the inborn errors of immunity. And we can see, for an example, here in 2017, we almost doubled the number of genes involved in this group. So this is more than a semantic classification, as I said before, but the way we see these patients, the way we are going to discover new genes and novel therapies is going to be impacted by this. And the name of immune dysregulation, it's not just, it's not just semantic, it's not just cosmetical, but if we see the 10 groups of patients of classification of inborn errors of immunity, the groups that has grown much more than the others in the last 4 to 5 years was especially those associated with immune dysregulation, especially the number of the group 4 and the autoinflammatory disease. So this change of nomenclature was responsible not just for identification of new genes, but especially these genes with immune dysregulation. And this probably is going to be the genes, the groups that's going to grow higher in the next years. Here, there is this new group. It was classified 2 years ago, that it was not in the past classification. And it's one of the groups of interest in our research group because it's not inherited and it's much more common in adults. So here in Brazil, we are working on the National Center for Inborn Errors of Immunity. We are trying to collect samples from all over Brazil. We are trying to put all them together in a biorepository, in a bio of patients to be followed for 10 years. We are trying to get the clinical information in the REDCap, and we are going to follow these patients for 10 years to see the natural history of involvement. But curiously, 50% of our cohort is composed of immune dysregulation. So it's also very interesting to see that the patients that were not diagnosed in Brazil that were sent to us was involved, or clearly, clinically, actually, we can say that they are within the group of immune dysregulation. So we see changing the years of the infections to autoimmunity and special inflammation. So another point that is interesting to observe is that we saw not just the expansion of genetics or in numbers, but the mechanism involved in the predisposition to infection, to autoimmunity and inflammation, the mechanisms that underlie these patients has also expanded in the last year. So we move it to this basic concept of biallelic was associated to loss of function. That is the classical model of inheritance of this genetics. We observe now several mechanisms of genetic mediated disease, which we can see the haploinsufficiency. We see the gain of function, as I said before, in the STAT1 patients. We see also these very interesting groups of haploinsufficiency and also dominant negative disease. And we are now observing that CNV, the copy number variations associated with these disorders, as is also an important mechanism involved in this predisposition. But all that we observe from the clinical point of view and from the phenotypic point of view, even at a cellular level, is not completely explained by the gene by themselves, because even within the same family, we have patients with the same genetic background and we see different phenotypes. So there's something more that we are not still able to explain, or we are not completely able to evaluate, that has to explain these differences between the same genetic background within the same family. So we are starting to study as second hit variants, for an example, as a possible modifier of the phenotype. We want to study, not study yet, but we want to study the exposure of this patient within Brazil. So we are trying to look if within the different geographically distribution in Brazil, we can have some explanation of the phenotype differences. And we already have some answers to why age and sex may also influence in the phenotypic variability, especially for the formation of somatic mutations that naturally occur within the age. So this is the work we want to move in the next years, to try to explain if this incomplete penetrance of the genetic background of these patients may also explain and also be transformed into therapy or orientation to these patients. So this is something that we are understanding now, that not all mutations can be inherited when we talk about inborn errors of immunity, because one of the concept that the physician has is that immunodeficiency or inborn errors of immunity are all derived from germline mutations. So this was a start point for us in the last years, in which we can see in the genetic sequencing the allele fraction of 50% or 100%. But after the discovery of one gene associated to adult onset disorders, that is the VEXA syndrome, we saw that not all disease, not all mutations should be inherited. They could be also derived from somatic mutations, from a small allele frequency of cells that could be responsible for the phenotype we were observing. So this changed completely the way we analyze the genetic data of these patients, because we moved from analyzing just germline cells to analyze also somatic mutations or somatic cells. So we are moving now to the high deep frequency sequencing of this genetic data to see very low frequency cells, especially in CAPS or ALPS genes that are related to the apoptosis, or in genes associated to adult onset disease. So this is the first change that happened in the last years and that we are working now in our patients. So this is the first VEXAS patients we studied. Actually, we did a collaboration with Alessandra. We tried to analyze the role of inflammasome in the formation of VEXAS syndrome. We could not see a clear difference among the patients and the controls, but this is the first disease that changed all the rules for the inborn errors of immunity, because it's not congenital. It initiates in the last decades of life, especially after the fourth. It's not germline, it's related to a somatic mutation. It is not systemic, it's restricted to a single hematopoietic lineage, and it's not a classical immune gene. This is a gene related to more metabolism than the immune function. So the same disease, there's one disease, changed all the concepts that we knew when we were talking about inborn errors of immunity. So this is very interesting also from the point of genetic analysis, because we came back to all the patients that we had sequenced all over these last 2 years, and we looked through all the exomes and genomes to see we have missed any other patient with VEXAS. And it's important that all the exomes and genomes analyzed were negative to UBA1 genes. So the exome, the classical exome and the classical genome cannot find it. So it was important for us to understand that we needed an additional genetic sequencing method to evaluate the somatic mutation. That's why we moved to this 2000 diff target sequencing to analyze all the genes that may be involved in somatic mosaicism or somatic mutations. And I don't know if everyone is aware, but probably they know. I think that everyone here may be familiar with Sanger sequencing, but with Sanger sequencing, that is the old technique of genetic sequencing, we are able to capture the 90% of this mutation that is located all in the same codon, that is the 41 in the exon 3 of the UBA1 gene. The problem is that it takes time, it takes money to understand, and it's very difficult to analyze. So it's not easy. We need a next generation sequencing to evaluate these somatic mutations. So these are the genes that we are working with somatic disease, and this is when you have to think also for somatic mosaicism or somatic mutation in patients suspected with inborn errors of immunity. But not all the disease, not all the disorders that we are talking here are caused by genetic alterations. So that is the group 10 of phenocopys that is listed now in the IUIS classification that are not genetic. So this is very important or interesting, because the genetic negative patient can also still have an inborn error of immunity, and it's not related to genetic background. It's related to antibody, anti-cytokines. So here there is a group of 6 cytokines that we have already studied. The most famous here is the anti-type 1 interferon that was famous when during the COVID-19 outbreak, because 10% of the patients that were severely affected with COVID-19, probably they had an antibody against interferon type 1. And also we are studying now the patients that get severely affected after dengue or chikungunya and other arbovirus. They may also have this severe this inborn autoimmunity that is an antibody against the type 1 interferon. We don't have this in our laboratory, but this is one of the fields that we want to study more now, is the antibody against cytokines, because this may explain not just the disease, but also different outcomes in the same disorder. There is one important group recently from New York. They studied this antibody against cytokines in the development or the outcomes of cancer. So it may also explain why some patients with the same cancer may have different outcomes, especially in the inflammatory pathways. So this is something that we have to look after in these patients with severely affected patients. So this is something that that we also have been trying to evaluate, because we are now analyzing just the exomic part of the sequencing. So 1 or 2% of the genetics that we analyze, it's what we are studying now. So 98% of the genome we are not analyzing, and they may have answers for what we have been looking for. So this is something that we have to work, and we are going to work in the next years, is to look to non-coding parts of genes, which is long numbers of amino acids that are the promoters and the UTRs of the genes that are not clearly there's no clear implication of what we see there and what we see in the phenotype. We still don't look to CNVs in patients with inborn errors of immunity. That is something that we have to go deep into the analysis. That probably there are many or several diseases involved in CNVs. We don't go too deep into intronic, because we have this rule that just 15 to 20 amino acids before the exonic part may have an impact, but different disease of metabolism or other disorders, deep intronic variants may also explain what we could see. We are not, we're looking still to repeated expansions that are some disorders that may affect the neurological involvement in patients with inborn errors. So this is something also. And we are not looking to hetero elements. That is something that may impact also, especially the interferonopathy. So although we have all the exome and genomes there, we are just studying until 2% of what we see. So we have several things to work on in the next years. So, and why we need this collaboration with to work in group, to work as group, especially to work with who work in the laboratory, because although sequencing give you a hypothesis of what a gene may cause, we still need to work on functional assays to prove what this gene causes in cell lineages and also in animal models, and what this is what's going on in the background or in the at the cellular level. So it's still necessary. Genetics is not the answer. What I want to say, genetics is just an hypothesis of what we have to go through. So it's very important to have these collaborations and to work as a group to move to this point that is the functional assays for all the disease. We started, I mean, I have done my PhD, and I did some specialization outside Brazil in autoinflammatory disease at the NIH and in Italy. I lived in Italy studying autoinflammatory disease, and I started working with autoinflammatory, but I recognized over the last years that even the points that I was studying was the same point that other groups, such as in the cancer, such as in the infection, was studying the same thing. So we need to sit to put together all our knowledge to combine our information. So we started working several years ago in the inflammasome-related disease. So this is the group of disease that I worked with Professor Alessandra. We worked initially with the patients with the NLRP3 mutated. So we worked trying to develop a functional assay to diagnose this patient, especially those that are negative. And we are trying now to work on a functional assay to map the gene involved in the familial Mediterranean fever. And recently, Seth Master proved that all these diseases are associated within the same machinery that is a mutation that works within the actinopathy. So they are all within the same phenotype. We are working in our laboratory in the School of Medicine, also in the Sigaz-Sting pathway, that is the pathway involved in disease that looks like lupus and are not lupus. They are Mendelian disease associated with this inability to distinguish what is self to non-self um, nucleic acid. So we are working with this group of disease. So we developed a signature to measure the interferon stimulated genes, and with this we can select the patients with more target phenotype. So we can select the patients with the SAVA copy interaction or the Caidy-Gutierrez are the two main groups, but we have several patients with a clear phenotype of interferonopathy, and the positive interferon signature in which we had not found the gene. And it was important in the last years because this concept allowed also the development of several drugs that are important for the inhibition of the interferon pathway, and many other drugs are coming in the next year for this group of disease. We are also working with the FOXP3, the IPEX FOXP3 pathway. A lot of patients came to us as autoinflammatory disease, and in the end we saw that they were IPEX, that is the immune dysregulation endocrinopathy X-linked disease. That is the gene responsible, the gene is FOXP3, responsible for the formation of the FOXP3 protein. That is the gene that won the Nobel Prize last year, I think. So we have been working with this disease, and it's very interesting that we found that the mutations located in this part of the gene are associated with DNA binding, and they have a less severe outcome when we look to the disease that has mutations. For an example, these two mutations in this part of the gene. This part of the gene doesn't bind with the DNA, and this is something very interesting because we were collaborating with Professor Rosa Baqueta in Stanford, and we found that this difference is also important for the outcomes that they were doing for the gene therapy. So there is an impact of also for the mutation, the location of the mutation, and also in the expression of the FOXP3. It's a paper that is coming out now in the next month probably, but we found some important difference in Brazilian patients with other patients, and this is one of the explanations, but maybe other factors may explain in the near future. So we are also working with the tuberculosis or diffuse tuberculosis with the patient in a group of collaborators with Africa. So we are looking to this axis of genes involved with diffuse mycobacteria disease. So this is also an important topic that we want to work in the next year, especially for the development of antibody against in each of these cytokines. So more than just the phenotype, it's another example here. It's a patient from Rondônia, from the state of Rondônia. He came to this dermatologist because of the allergy, just because of the skin allergy. And he looked through the skin allergy and he found several virus infections in the skin of this patient. He also had some recurrent pneumonia, but here some lung damage because of the recurrent pneumonia. And we found that this patient has a novel, not yet reported mutation in a cytoskeleton, in the region of a cytoskeleton. So we found a novel mutation that lies within the DOCK8 genes, but has also an impact on STAT3 and also an impact on the VASC gene. So it's very interesting to observe that the same phenotype of allergy or something very common that is allergy may have a different genetic background. So that this categorization or this classification of 10 groups of disease in the IUIS, it's not something semantical, it's not just something cosmetic. It's very important for us physicians and for those who are studying to try to understand the pathways of involved in these genes. What we are observing at that all these patients, they share the same need or the same therapy. So we were observing the last years that the same drug developed for the autoinflammatory disease, they may also treat almost all the autoinflammatory disease. So different genes may also be involved within the same uh, mechanism of these disorders. The expansion that we are doing here in Brazil, it's not just because we wanted to analyze our data, but we are seeing something necessary for the analysis of the whole world when we talk about genetics, because the whole world needed to have more data to compare, to have more data to really be sure of what is rare, what is not rare. And the Brazilian population is fundamental for this genetic data, because we have at least I was doing a research with a colleague from Pernambuco, and we found at least 7 different populations that were responsible for the formation of our population. So we have from one side the Italians or the Europeans, let's say, that comes through the littoral here in Brazil. We have from the other side the Spanish and the indigenous that comes from the other part of the Brazil. And we have the real Brazilians that are the ones who lives in the center of Brazil. So they are not the same genetically, so they are different, and it's necessary to understand what is the background of this population to see what we can have to align in the genetic background. So this is the project. This is just a slide of what we are doing. We created this multi-center study that calls CNE3Z, National Center for Inborn Errors of Immunity and Immunity Regulation. That is the principal investigator is Professor Magda. That is, she's online, I think. They received 40 million reais to do this project. So what we are doing is registering this patient in REDCap. We are doing not a biobank, we are doing a biorepository of samples with RNA, DNA, serum of all these patients, and we are trying to integrate genomic data to analyze both together. So that's what I'm doing. That's why I'm overwhelmed with many things in the last years to work on this project, and it's working very well. We have now 74 centers all over Brazil, and I was talking to Diego because we want to create a discipline to integrate all the post-graduation programs. So this is an example of something I want to do there in our laboratory. We have up to 3,000 samples of patients from all over Brazil with different type of disease. Half of them, we could find any genetic data that could explain, but genetic data that clearly explain everything just in 10% of these patients. So 90% of these patients, we don't have a clue of what's going on. We just have some hypotheses. We need to prove many things. We have sequenced already 1,800 of these patients, and we have analyzed just 1,000. So we still have a lot of things to do, and we have many data that you can also, everyone can are invited to work with us. We have all these patients and all these centers, they come to us through COBE. That is it's not a medical, it's like a community that I am the president. So we monthly have meetings online to give back the lectures with what we are learning in the laboratory to the physicians from all over Brazil. So each month is a physician from one of the centers that come to us online to talk about their cases and all the genetics that we are studying, and what the genetics has impacted on the care of these patients. We are also doing something very interesting that I realized when I got the Kobe, that all the knowledge was centered in a single place and in a single person. So every time we were doing the Kobe network, it was also always the same person that was with us. So we decided to go to the place where this person work and to invite all the physicians, all the rheumatologists, dermatologists, infectologists, to improve this this network and the capture of these patients. And it worked very well. We have proved statistically that this Kobe network had worked for um, the capture, let's say, to receive this sample. So this is something that we are doing also that works, and it's something that everyone is also invited to not to go, but if they want to receive Kobe, we are able to go to whatever whatever they call us. So these are the most frequent genes we have found by now. So the most frequent gene is the TACI, the responsible for the B cell maturation. And the second, third, and fourth genes we found is associated to autoinflammatory disease. But this is something because of my specialty, because I was capturing patients with autoinflammatory disease, we are observing a switch in the genes responsible for immunodeficiency, the clear immunodeficiency, and this predisposition of infection. And it's very interesting that we see some differences when we talk about the genetic background of autoinflammatory disease here in Brazil and what they see in North America and also in Europe. This is very interesting also because we have found some founder genes for the Brazilian population. In two scenarios, clinical scenarios, we found that in two diseases, especially for DIRA, the IL-1 receptor antagonist, and for the Achaeus Gutierrez, all patients in Brazil, they have the same mutations. So it is a founded effect within Brazil, so it probably came with the same family from Europe that came to Brazil and were spreading the mutation in Brazil. So it has, and this is very interesting also when we are talking about testing, because as we know that this mutation is responsible for DIRA, we don't have to go to the next generation sequence. We can do just a Sanger sequencing for those both diseases that we go through the genes. We are also observing here in Brazil, I'm finishing now, that, for an example, the example of pyrinopathy. So the genes related to MFV, or the Mediterranean fever gene, they are classified now as pyrinopathy. So genes, disorders associated with the pyrin gene, and not all the mutations are, I still have already have a phenotype associated. And we by analyzing all the genetic data here in our cohort, we have found 13 new mutations in places not yet described within the pyrin gene. So we have 13 patients with an unexplained disease in which we cannot explain already the mutations that we found, and probably we are proposing that we may have a new classification, a new phenotype associated with a mutation that only exists here in Brazil in the pyrin gene. So this is something that we are working also in our cohort. It is impossible to do this alone, so this is our collaborators. I'm trying to get people from all over the world, from all over Brazil, to work together, because it is impossible to do everything alone. So we have collaborations with Instituto Imagine to work in the interferon-mediated disease. We have collaborations with China in a project to work on the transcriptomics, especially, and at Stanford, and also with the guys from Africa. So in the end, we want to go back to the bedside. So I've moved it from the clinical to the laboratory, and now we want to go back to the bedside to give possibilities, therapeutic possibilities to these patients. So this is something also that we are working. So Up to now, we have some biological drugs to treat all, especially autoinflammatory disease, but they are very expensive and they have to take it for a lifetime. So it's like a lifetime treatment for the disorder. And several of these diseases still don't have 100% remission even when they are treated. And if this treatment doesn't work, we have to go to the bone marrow transplantation, which is risky. It's the rate of death, it's high, and it's very difficult to get because there is a line to go in the public system. So not all the patients can have the bone marrow transplantation. So probably the future of this disease is gene therapy, and this is something that we have been working also, especially with the IPEX, in collaboration with Professor Rosa. I don't have the pictures here, but we have two patients with IPEX, that is the disease caused by the FOXP3, that they were treated in this the clinical trial for IPEX. So one of the patients, it's within the DNA binding, and another patient is within the non-DNA binding. And the patient that was treated in the DNA binding is very well, not taking anything for up to 2 years now. He's not taking any medicine. So it's proof of concept that this gene therapy really works for this patient. But the patient that in the DNA binding, non-DNA binding, unfortunately, he's not doing well. He was sent to bone marrow transplantation. So although we have some answer, we still have to work more to have a better therapy for all these patients. And this is the gene therapy for FOXP3, but we have 500 genes that we have to work for the development of this disease. And going back to the disease of IL-1 mutated patients, so this is DIRA, the deficiency of the natural antagonist of the interleukin-1. So this is not infection. This is the absence of the protein that binds to the receptor of interleukin-1. So without this protein, these patients have this loop of IL-1 production, and it resembles sepsis. That's why these patients were treated as sepsis. And after the genetic sequencing, we have gave this patient, all of them, we blocked the IL-1, and they are doing very well since then. So this is the fit of the patient I see at that time. So although he's alive, he doesn't have some fingers here because the osteomyelitis has eaten his fingers. But which is I think what is from the genetic point of view very interesting is that all of them has the same mutation, and we found one patient in Rio Grande do Norte, 2 patients here in Minas Gerais, 1 in Santa Catarina, and 5 here in São Paulo. So where the first came from, we still don't know if they came here in the Northeast or they came here in the Southeast or in the South to spread the same mutation all over Brazil, but probably they all came from the same guy that probably transferred from, I don't think, maybe from Europe. And this is also something that we published last week. When treated, all the patients survived. When not treated or not recognized, all the patients died. So recognition is something very important. So that's why we need to talk to physicians to recognize this disease, because if they got recognized, diagnosed, and treated, they are still alive, even without some fingers, but they're still alive, doing very well. So these are my take-home message. So inborn errors of immunity, they are the natural genetic experiment in nature. So we have to study these patients. We have found that now 500 genes, but we probably have more than 600 in the next classification. Every 2 years we have this classification. We still need to expand the phenotype, the mechanisms, and the technologies used to this disease. And I have shown so many aspects in which we need to work, and everyone here is invited to work with us. We have moved the bottleneck of this disease. So before was the recognition, now we have to not just recognize, but we also have to prove the genes that we are finding, to understand the mechanism, to understand the difference in the phenotype. So we still have many functional necessities for these patients. always think about the somatic mosaicism. So if you do not find a mutation in germline mutation, think of somatic mosaicism or other phenocopies. That's something very important also to this disorder. And diversity in population is necessary. So we still need to work to have more genetic data. So if you want to get involved, we are more than open to receive students, to receive researchers, ideas, everything that want to come to work with us. We are open to this. And this is our group. Our leaders are Professor Jorge Calil and Magda Sampaio. Jorge from the adult department and Magda from the children department. And this is the undergraduated, the postgraduate student from our department. So this is a huge group that we have working with us, but everyone is invited to join us. So, and I also want to thank all the collaborators, the families, especially the patients and the families that collaborate with us. This is my email. Who wants to send me email and to talk? That's what we are doing. So thanks. I'm open to questions now.
Deixa eu ver. A Steph tinha marcado uma reunião aqui.
Mas eu tô saindo, tô terminando.
Só por curiosidade.
Não, ela tá aí, ela tá nessa sala, minha querida. Tá bem aqui. Oi.
So, thank you very much.
Congratulations. Thank you so much. Wonderful lecture. Congratulations, Gustavo, for the organization of this meeting. Congratulations, Leo, for the outstanding lecture.
Thanks, Professor.
So thank you, Leonardo, again for this talk. It was amazing. So we usually open for questions for the students first. So if anybody has a question here or in Rio or in the chat.
No, I don't think we have questions.
Okay, so Bruno, have a question there?
We don't have questions from students here, but we do have a question from one of the faculty.
So please go ahead, because I will have a question after that too, and Gustavo.
Sure.
I have a question also. If
Please go ahead.
Professor Marcelo, so
Yeah.
you can ask your question after Roberta, please.
Sure.
Okay, thank you. Roberta, please.
Thank you.
Hi, I don't know if we are not seeing them anymore.
No, that's okay.
Is that okay?
You can just go ahead, yeah.
Hello, very nice talk. Thank you very much. I don't know if you guys hear us over there.
Yes.
So I also work with inborn errors of immunity, and I was wondering, how do you do your ACMG classification? And are you looking at all to structural variants? Because this can be challenging, right?
To structure, we are not looking to structure variants now. Because we received like several demands over the last year, so we prioritized something to do. So first, what I was explaining before, first we are looking to the known genes to give the results to the physicians. So that we are looking. So first we clinically classify these patients within these 10 groups, because we are physicians, so we easily can see the clinical picture and to classify them within the 10 groups. And then we look through these groups if we have any known gene causative of this disease, or if we have a gene that is not known, but it's really important, let's say with the in silico classification very important, or with not with zero frequency, for an example, in genes. So we look for first clinically, then to the genes within this panel. If we do not find, and if the clinical phenotype of these patients is maybe associated with several other rare disease, we go back and we look through the genes as a rare disease or unknown disease. We analyze them as a rare disease per se. So we look to all the recessive genes, all the compounded heterozygous, all the autosomal dominant, and all the X-linked. As by now we know that all the disorders within the inborn errors of immunity are not associated with CNV. We don't give that much look through the CNVs. We look to the knowns like the 21 key, we look to this, but it's something that we have to work with in the next years. It's look through these mutations.
Okay, and how do you do your CMG classification? Do you use like a software?
Yeah, because of the project, we bought the NovaSec X, the Illumina NovaSec X to the laboratory, and with the NovaSec X, it comes with the Imagine, the Illumina Imagine analyzer. So it's very friendly and friendly to use. So it comes with the ICMG classification by itself. We just changed the ICMG classifications because we can reclassify within the platform. So, and if you, for example, you use the same platform, we can share the networks to see if you have seen the same mutation, and if we see the same mutation, and we can reclassify it together. So, but we use the Imagine platform to classify the ICMG and to reclassify the ICMG.
Okay, thank you very much.
You're welcome.
All right, so Dr. Marcelo Barcinski was wishing to ask a question. Is Marcelo still on?
Hello?
Yes.
Yeah. Well, congratulations for your lecture. Yeah. I want to ask if you have, perhaps I lost it, but if you have X-linked mutations, and if so, if there are differences between males and females, and also if the X inactivation is the preferentially of the mutated X, as it is in other situations.
Yes, we have some disease that are X-linked disease. So we have some SCID phenotypes, so severe combined immunodeficiency phenotype. The main mutation associated with this group of disease are genes in the X gene. But as there is the newborn screening in Brazil, we don't have these patients in our cohort because they are going through the newborn screening group, let's say, like something like this. We have the VEXAS, the disease, the adult onset disease that I showed you. It's associated with the X, is X-linked, it's a gene in the X gene. And we have some females with the mutation in the X gene that are also affected. We have not gone too deep to analyze the differences between the negativation of one gene in X in another gene, but I think that this may be an answer to why we don't have much more women affected with the VEXA syndrome, and so just some women.
Thank you very much.
You're welcome.
All right, so Gustavo, Bruno, you want to ask a question?
Sure.
Go ahead, Bruno.
Thank you very much, Leonardo. It was a really very nice talk. I was wondering, because it's not really my cup of tea, dealing with the patients, right? But what is the flow chart for these analyses? The patients are coming, they are all sequenced. And how do you decide that if you follow through, if there is no known mutation, how do you decide what you do next? Is there any other test that may inform what is the next step? So just for me to give
the flow chart,
a fuller picture of how these things are done.
So if a patient comes to us and if it has already been sequenced, for an example, in exomes in private companies, or previously we have been sequenced through genome somewhere. First, we do the reanalysis of this. So they send to us the FASTQ or the band file, and we do the reanalysis first. If it has not been sequenced, and if it's a clear known inborn errors of immunity, for an example, we go through exome, because probably this is an exonic mutation. And if it's not clear, we try to put these patients in a genome sequencing, or to look more for sequencing that we have like larger files to see, because of the possibility to reanalyze also this disease with not clear phenotype. So this is the first step that we do to decide in which kind of sequencing we're going to put them. Or if they are, for an example, DITA or Arcaide Gutierrez, that we already know that they are founder effects, we go directly through the Sanger of this part because it's easier for us. And for VEXAS, that we need some deep sequencing, and we don't have it now, we go directly through the target sequencing of the Sanger. So this is how we decide initially. Then we are trying to develop assays to classify clearly these patients into some groups. For an example, with Alessandra, we developed this inflammation-mediated assay. We have this activity of the inflammation activity, because if we have a proof that these patients are inflammation-mediated disease, we put them into the inflammation-mediated disease. Or if they are interferon positive, they go on through interferon signature. So we are trying to organize them first in clinical, then in cell phenotype, and then we go through the genetic analysis of these patients. In the end, they all got the result like this for us. Positive, clear positive, we don't need anything else. We are going just to follow these patients for 10 years. Every 5 years, we want to collect clinical data to see differences in the phenotype. And if it's probably, so we don't have a positive result, but we have a possibility in the genetic background. So we need some we need functional assays to reclassify, to do whatever we want with these patients. And we call them as negative when we don't have anything, not in the cellular level, not in the genetic level. So this negative, it's something that we are going to reanalyze time by time, or time by dependency of what they are asking us. So this is our flow chart of analysis and interpretation.
And for the anti-cytokine antibodies patients, how do you get to them? That's what I was really curious about. How do you decide to look for the anti-cytokine antibodies?
We don't have yet the anti-cytokine assay in our laboratory. We do this in collaboration with Jean-Laurent Casanova. So what we do now is, we have a group of disease that we clinically recognize that they have probably an anti-cytokine. So we have, I think, 100 and something patients with severe dengue, severe something, and they are all genetic negative in the germline. This is the first thing we did. We are going to put them in the deep sequencing to see if they have any somatic mutation, the second step. And if they are clearly negative to this, we are going to send all them to anti-cytokines, because there is a student doing this, so this is the step she's going to do.
Right.
We don't have the assay now.
Sure. Thank you very much.
Great. Gustavo has a question.
Actually, first of all, congratulations, beautiful work, and organizing this is not a simple task. We know. Now, just following Bruno's, I was just curious why you guys consider this antibody-dependent anti-cytokine as an inborn error.
This is a critical question to the classification, because these are not inborn, not the somatic nor the anti-cytokine. So I think they try to insert this in the classification, but for sure this is not inborn. They are just mimicking the phenotype that we observe in the germline, but by book they are not inborn errors of immunity.
Very good. So I have a question, actually, more of a, maybe it's out of the scope, but something that I was thinking about. So first of all, congratulations for bringing up genetic classification of whole Brazilian population. So this is really nice, right? So mixed, and there's so much thing that can be related to infectious diseases or whatever, autoimmunity, autoinflammatory diseases. And I was thinking, if there is anything that you have already verified or identified in Brazilian population, especially from, native populations, that can be associated with differences in susceptibilities to neglected diseases that are present here.
We don't have that much in our cohort. We don't have that much native. Probably we have some mixed from Mato Grosso do Sul, from Amazonas, from Roraima, from that part. We may have some mixed because we don't have the authorization to get the DNA from the indigenous. We don't have this ethical authorization. But sometimes when they are mixed, they send to us to genetic sequence. We have not analyzed this difference because we cannot be sure who is native, who is not in nature. I mean, it's self-declared. But we have been looking through slaves, descendant from slaves, and non-descendant from slaves. So we have some differences. We got some genetic background of this from Bahia and from that region. For an example, they are self-declared as slaves descendants, and from São Paulo that are clearly Italians population. So we see some difference in the background of these patients, but we still need more. The number have to grow to have a more robust statistical analysis of this difference. But when we see through some genes, we find some variants that are really frequent in Bahia and are absent in São Paulo, for an example, or frequent in the south of Brazil, also from German descendant patients. We also see some variants that are present there, and they're absent, for example, in Goiás, that they don't have there. So we can say who is from where when we look through this genetic background. Probably in the next years, we are going to have more clear picture of whole Brazil.
Great.
That's why I said probably from Goiás, Mato Grosso do Sul, not Mato Grosso, but Mato Grosso do Sul, Goiás, and probably Tocantins. They are really Brazilian. They are the mixed population of everything.
So one thing that comes to my mind is like, we always hear in history books that the indigenous people were more susceptible to cold viruses and stuff like that, killed them a lot just by contact.
I didn't know that.
Yeah, and many people consider this to be just a polymorphism from MHC molecules that, and I don't know if there is anything that could be related to any kind of inborn error. I don't know.
I think so, especially because they have a high rate of consanguinity. So probably what we are going to see now, who is alive, it's the segregation of someone that had something in the beginning and that has not transmitted to what we are seeing now. So probably we are not going to see everything, but we are going to see some differences.
Very good. Very good. I don't think we have any more questions, but thank you very much again, Leonardo. It was very good. It was amazing.